All right, hello everyone. Welcome to the webinar. See people entering. We'll give just a minute here for everyone to get settled. All right, we'll kick things off here with our webinar. Hello, and thank you for joining today for Transforming Turf, Breakthroughs in Golf Course Health with Replenish Nutrients and BeCrop. I'm Sunny Kaercher, Account Manager at Biome Makers, and I'm joined today by Gus Plamann, our Senior Technical Agronomist at Biome Makers, Mike Hamilton, the founder and CEO of Turf Dietitian, and Kevin Erickson, the Chief Operating Officer at Replenish Nutrients. We have a fun topic and a lot of content to cover today, so I'm going to ask each of our speakers to share a bit of their background and about their companies as we pass the mic around here. So our crop of focus, turfgrass, is unique in many ways. First off, recreational turf is highly controlled. From mowing schedules to custom blended sand-dominated soil mixes, it's about as far away from a natural system that we see in outdoor commercial cultivation. And secondly, instead of yield being the conventional indicator of success, success is measured by aesthetics and performance. So how does the course look? How well does a ball bounce or roll on it? And considering this, superintendents are under high pressure and they have very little room for error in their management decisions. And so though it might seem like a challenging place to introduce biologicals, our speakers have some valuable insights on how and why turf can be maintained with biology. So a little bit of our roadmap for the day. First, Mike's going to share on his experiences in golf course management and his preferred approaches to integrating biological products and practices, as well as his learnings from using BeCrop for diagnostics. And then next, Kevin will share about Replenish Nutrients' approach and application of their phosphorus solubilizing product Rebuilder. And then Gus will share the results from a BeCrop trial conducted with Rebuilder at a Montana golf course. And then finally, Kevin and Mike are going to tie it all together in an open conversation about practical insights and applications of all this information. And we will have a Q&A at the end, so please feel free to drop your questions in the chat or the question box. You'll see both of them there as they come up. We'd love to hear what's on your mind. And so for those of you new to joining us, Biome Makers decodes soil biology. We do this by combining advanced genomics, machine learning, and microbial network analysis. Our technology, called BeCrop, is an agronomic tool with many uses, including product research and development, commercialization support, in-field diagnostics, and soil health measurement for corporate and consumer reporting. And our team is committed to connecting soil biology to agricultural decision-making to optimize practices and reverse the degradation of arable soils. So we see microbes as reliable bioindicators of soil health. And given that a teaspoon of soil can have over 10 billion organisms in it, soil is very much alive, and it plays a critical role in plant health and ecosystem balance of any cropping system. BeCrop Technology translates these complex soil microbiome data sets into functional indices. So these indexes give us a view underground, allowing us not only to see what is invisible to the human eye, but more importantly, understand and take action based on the biological strengths and or shortcomings of any given field, or in today's case, a golf course. So without further ado, I'm going to tee up Mike Hamilton.
Hello, I'm Mike Hamilton, and our company is called Turf Dietitian. I was a golf course superintendent for many years, and then in between superintendent jobs, I went to work for a microbiology company. Got introduced then, and that was about 30 years ago, when you'd walk into a maintenance facility and talk to the superintendent and walk out and they're laughing at you. They didn't know what microbes were, most of them. I mean, yes, they did know what they were, but they didn't know how to use that community of organisms in that ground. So am I switching slides, or are you guys switching them? Okay. Gotcha. I am going to rush through this because I'm This is a 10-minute presentation, and usually this is about a 4-hour presentation for us. So, but microbes are involved right from the beginning, right when the seed starts to germinate, or even they assist in the germination of the seed by producing enzymes to break down the coating. Then you've got these molecules that stick together and they form uh, this photosynthesis equation, with the net result of this first part of this process is the formation of chlorophyll. And you have to have microbes in the ground to do that. And, you know, in turf, the word anaerobic bacteria is kind of a bad name, but we need these green and purple sulfur bacteria to complete the photosynthesis process. But what amazes me, not only that chlorophyll molecule, but when we look at amino acids and proteins, very complex chains of nutrients and those particular acids. And oops, sorry, I keep pushing my button, Sonny. There you go. Now I got distracted. Sorry about that. So anyway, a natural Bermuda grass plant or bent grass plant, which is the majority of the golf courses in the country, use those two species. If you allow that plant to grow in the wild like this, it's as healthy as it can be most of the time. Sure, there's death in these turf grasses. They don't live forever, just like humans, but there's no input into these photos. These are just filled of common Bermuda grass growing and bent grass growing. And the reason that happens is they can produce enough sugar and food to feed those microbes in the ground, and the microbes in the ground are going to take care of that plant. All the needs for that plant, whether it's under attack by a pathogen or it needs a potassium molecule, there's a tremendous communication going on. So this all sounds very simple, but each step of the way you go, there are thousands and thousands of factors that affect that. Ooh, button. And here's where it goes wrong on golf course, it's that mower. And there you go. So we're cutting now. I'm hearing heights of a tenth of an inch, an eighth of an inch. So that plant can't produce enough sugar to sustain that plant without chemical usage. And so you have to build an artificial environment down there by using microbes and food sources so that there's enough of them down there to defend that plant when it's in stress. And when I came across the BeCrop report, that was the kind of the last piece of the puzzle. I mean, I hope we still get pieces to the puzzle, but we test for amino acids, plant growth hormones, carbohydrates, nutrients, everything in a plant we can test for. We can test for nutrients in soil, but we've never been able to test for microbes. The older tests were very, very expensive. And when you got the report back, it was nowhere near as detailed as what we see with the BeCrop report. It's addressing the stress factors and what organisms are there to protect that plant and adapt that plant to stress. Next slide. So this is a golf course that superintendent said, you'll never get me off of fungicides. Everybody's on a preventative fungicide program anymore. You'll never get me off. So he gave us this green. This is a practice green. And this was after 9 months into the season, and not one chemical was sprayed on this. Now, he had been using biological product for a while, but I asked him if he'd cut out the chemicals, and this is the end of the season, and that turf is fantastic, as good or even better, is definitely healthier. Not a big visual difference between that green and the other greens, but to be able to do that without the use of chemicals is huge. And Do I suggest you start using this stuff and are using biologicals and just ripping the Band-Aid off and say, I'm not going to apply any more herbicides or fungicides? You can't do that. You have to slowly work your way up to where you're not so dependent on those type of products. Next slide. So this is just a test we did, and we put it on a sheet where we did all the totals. That's the BeCrop report. The numbers, the higher the number, the better that particular factor is. And in the beginning, we were just trying to work to build up the microbial populations, because first of all, you're cutting the turf at 1/10 of an inch. Second, it's sand that they'll throw 10 to 20% organic, and usually about 10% organic into those sands. So you have no microbial activity. So this was a new green, and so we knew we were starting from scratch, and we could build that population the way we needed to build that population. And this is a bentgrass green in Pennsylvania, and those guys live by fungicides, and so we were really working on building the population. You can see when he started back in January. Of course, it's cold in January. I take that. That wasn't January. I'm sorry. April. He was in April. He had 1 billion bacteria, 1 million fungi. We got to the end of the summer. He had 10 billion bacteria and 10 million fungi, and the endo-ecto ratio should be 1 to 1. He was closer to that. Now, he was the first course that I got to try products that had any endomycorrhizal from golf course soils. Usually every one we get, there are no endomycorrhizal in there. And when these guys start seeing that change is when that endomycorrhizal gets into that picture right there. So he was even over at 3.3 to 1 at the end. Ideal is 1 to 1. But what was really impressive is changing that ratio of those fungi to bacteria, 1 to 4,000 down to 1 to 149. So big changes. And you can look at this, and there's not everything didn't change, everything didn't go up, but the majority of all these categories went up, which, next slide. And this is the nutrient portion. When you look at these types of report, you can't think that, oh, it says I'm low in nitrogen, I'm low in carbon, I'm low in this. No, these are the bugs that are creating the pathways for these nutrients to get to the plant. And so if it says low, it doesn't mean, oh, I got to go out and apply fertilizer. It just means that you need to microbial populations in there that are specifically designed for that nutrient you're having a problem with, and get it to the root zone. And then those endo and ectomycorrhizal will start dispersing everything that plant needs within that plant. Next slide. So carbohydrates, like I said, we started there. And this is what's amazing to me when you look at some of them, fructose or any of them. And go to the next slide. So these are amino acids. And you see the top part of that amino acid is the glutamic acid. And the other, I apologize, I caught a cold the other day. The shaded areas are showing the side chains. And the side chains is what gives those amino acids their individuality. And what amazes me is they're built the same way every time. You get proteins. Go to the next slide. Well, those are plant growth hormones, but you'll get protein development that might look like a snake, and it might have 200 amino acids on it to form one protein, but it does it the same way every time. And how does it do it? Connecting positive charges to negative charges. But if you were to throw a handful of magnets, positive charge, and a handful of negative charge magnets in a a barrel and painted them all different colors, you could throw them in there and you would spend probably 20 years before you ever repeated that. And what amazes me about soil biology is that these things are put together the same way every time. So that shows me that there isn't an intelligence process going on in that plant, in that soil. And we all know that we wouldn't have any life on Earth if it weren't for microbes. So, but amino acids is the last thing. I mean, plant growth hormones is the last thing we've added to what we do, because those are all the precursors for stress-related problems in the plant. So we think we're going to get a lot of value out of that part of what we do, and then having the ability to use BeCrop tests to put all these pieces of the puzzle together is just fantastic for us. Next slide. So use bugs. Next slide. Not drugs. Next slide. And then this is the last slide. And these are just some of the books that inspired me. The Healthy Crops: A Revolution in Agriculture are, that's a book that was written in the 1950s. And it was from a scientist who had bought into the whole chemical expansion and we got to use chemicals for things. And he instantly found out about 2 months after spraying chemicals that, yes, the target pest went away, but the next thing that came in were mites, and they couldn't control them at that point. So he instantly started looking at healthier ways to grow plants. But I do recommend reading up on it if you like the science part of growing grass and turf grass, or parks and recreation guys. If you like the science part of it, start here. Start reading these books, because it'll just open your mind up like it's never been opened before. It's so beautiful and simple what goes on in that soil, and then we got us, man, coming along, and we're the ones that are causing all these problems, and so there are ways for us to get around ourselves. That's it. And I apologize. I feel like I missed a million things, because, like I said, this is usually a 90 slide, 4-hour presentation. But I couldn't do what I'm doing now without the BeCrop test. It's just fantastic.
Thank you, Mike. We'll have some time at the end to tie it all together too.
That's it.
All right. Again, so Kevin Erickson, I'm one of the founders of Replenish Nutrients. Myself, Neil Weens, and Gerard Philpott, back in 2018, developed a company. So you want to go to the next slide, Sonny? I'll give a little background. So again, our main goal was to develop a sustainable alternative to synthetic fertilizers, and really focused on the soil health and biology, just like Mike was saying. So our main goal was to develop products that would enhance soil health and increase the biology of the soil. So that's really where we've been focused on, and where we're going. So we are a publicly traded company. So again, if you're interested in us, it's a couple of the different areas you can invest in with us on the site there. So, or go to our website is the other place you can find information on us and our products. So next slide, Sonny. So, what our products are really mineral-based products. So with our process, we've developed a patented process where we're micronizing and using all mineral-based. So we've got 100% utilization of all our nutrients. We have no waste products that come off of our process. The key components that we're utilizing is phosphorus, potassium, sulfur, numerous micronutrients that are part of some of the products that we use. And then we're utilizing organic matter with live microbes. And I guess one of the key things that we do that's quite different is that we keep the biology alive in our manufacturing process. So all our granules have live biology, incorporated into them. And one of the things that we found is that our biology stays alive for up to 3 years in our granule, which is quite unique compared to a lot of products on the market. A lot of the products, I mean, you apply them to the soil directly or seed coat, but the survivability of a lot of those microbes is limited. So it is quite unique in what process we've come up with and how we've created these products. So next slide. So here's kind of the 3 products that we've developed since we started the company. Rebuilder being our first one that we'll kind of focus on, that we've utilized at the golf course. Another one is Hesso, which is kind of more of a maintenance product, and then Super KS, which is predominantly potassium based. So those are kind of our 3 main products. Next one, Sonny. So the Rebuilder product. So one of the key things in our product is that we don't have any nitrogen in any of our products, but they are all blendable with other fertilizers. I know nitrogen is some of the key nutrients for the golf course, and so we don't have any of that, but our products are quite blendable with other nutrients. With the Rebuilder product, it's predominantly a phosphorus and sulfur based product. And one of the key things that we focused on for the golf course, at least a lot of the ones in Canada have been banned from using phosphorus based fertilizers in some cities and a lot of the golf courses. So this product we really developed to fill that niche where we're not getting any leaching like you do with some of the fertilizers. So you're not having any algae blooms and stuff like that are at risk for waterways and ponds and lakes. So this product has a low salt index. It's full of micronutrients as well. So we've got copper, boron, calcium, magnesium, a lot of the balanced nutrition that is required. So next slide. So gonna get into what we did for testing. I mean, one of the key things that I'd like to, thank the Indian Springs Golf Course, Troy Truman, the general manager, and Benji, for the superintendent there, for helping us implement and do some testing. Like Mike said, a lot of golf courses are very reluctant to do any testing with kind of biological products. And Benji, Cheney, and Troy have been very, very good partners for us and have given us the opportunity to do a full fairway of testing on our products. So one of the picture on the left there is the fairway 14 that Benji was having issues with water infiltration and water holding capacity on that. And some of the reasoning when they developed that golf course, he said the heavy equipment ran up and down that fairway, and so they've got severe compaction on that fairway. So again, the soil sample to the right kind of shows there's maybe 2 inches of topsoil on that fairway. The other fairway that we used as a control, number 10 on the golf course, has probably 6 inches plus of topsoil. So quite a difference in the soil profile on that fairway. So our goal was long term. When we talked with Benji, it was going to be probably 3 to 4 years before we saw any results on the improvement. And again, that was pretty much how it went. The first couple of years that we looked at the fairway and stuff, we didn't see much improvement. A little bit the third year, started to see some improvement. And then this past year, we saw a considerable improvement in the water holding capacity. And that was due to a rain event in August where they had 1 3/4 inches of rain within 24 hours. And when we went out and looked at that fairway, there's really no visible signs of any leaching or runoff on that fairway. And when you walked on it, it was like a sponge. So again, all that moisture and everything had infiltrated into the soil profile. And he wasn't losing any of that moisture. So again, you know, the key to what we're hoping to do is try and reduce some of that risk of any chemicals running off. They've got a beautiful creek running through this property and lots of ponds. So again, it's making sure that all the moisture and nutrients that go on the fairway are being utilized and staying where they're put down. So with our products, they release over the growing season. So again, it's all biologically activated. So the nutrients are being released throughout the year when your grass is growing and needing those nutrients. So again, that's all biologically based. So you've got insect and disease suppression. So like as Mike talked about, I think, you know, one of the things that we're seeing that's, you know, kind of a side benefit from our products that we really didn't focus on or think about when we were developing them is that we're getting some insect and disease suppression with our product. They've got some grasshopper issues on some of the fairways at the golf course. And with this fairway, there's absolutely none on this fairway. So, and we've seen that in all our agriculture studies and stuff we've done with some farmers. They're seeing the same thing. They've reduced their pest control, so they're not spraying for grasshoppers. They've got less fungicides that they're having to utilize in the cropping system. So again, that kind of holds true for what you were saying, is some of the benefits that you're seeing with the biological activity increasing. We're seeing some of those results as well with our products. So again, a lot of the stuff that we're doing, you know, again, we didn't impact the golf course. Thankfully, it was nice and green. The fairway looks great. So we know how much of a risk that Benji and Troy took to have us do this main test on, and we really appreciate all their efforts. So with that, I mean, I'll maybe turn over to Gus, and you can go through some of the details on the testing that we did in the summer with you guys on this.
Yeah, thanks, Kevin. Really appreciate all the details and description from the field, and excited now to kind of tie this all together with some of our BeCrop data. First, just a really quick introduction. I'm Gus Plamann, a senior technical agronomist with Biome Makers. My background is in soil health, agroecology, and looking at sort of the biological component or aspect of agriculture. And at Biome Makers, of course, we've touched a little bit on the applications of our testing, and you know, Mike went over some of the results that he's used in his programs, and we really, the data that we're going to go through is actually from we're going to touch on a case study from this golf course trial that Kevin just went over and see some of the changes we saw in the soil biology that drove these outcomes as far as water infiltration, pest levels, overall vigor and you know, and growth of the turf on that treated section. So, but first, before we get into the details of the case study results, we're just going to go over a quick intro into Biome Makers technology for anyone joining today who's unfamiliar with us, and then a rundown on BeCrop Trials, the specific project and offering with our technology that was leveraged in the case with Replenish Nutrients on the Indian Springs Golf Course. And we've done a lot of previous work with Replenish Nutrients outside of the turfgrass space, mainly in large system row crops. And we'll tie in also some of the similar common benefits that we've seen in crops like canola, lentil, and other large row crops that we also were able to see with Rebuilder, specifically in this case in the turfgrass application. So for starters, on this slide, we just have a really basic overview of our analysis process at Biome Makers. As a soil biology technology company, we've pioneered a technique to confidently measure soil biological functionality. And this I like to boil this down to a simple two-step process to explain how we're able to take soil and generate data on biological functionality. So when we operate a flagship lab out of California and have partnership labs across the US and other international geographies as well as a lab that in Spain. And when we receive soil samples, either at our lab or a partnership lab, the first step is to generate biodata based on the microbial populations present in the sample. So our lab team uses a proprietary process to extract and amplify the DNA presence in the soil, and then identify through genomic sequencing what species are there and how abundant they are. So that takes us from the soil to the biodata, the second sort of step in the process. And that biodata is, you know, there's a ton of data there on microbial species, taxonomy, and abundance. But of course, to put that into agronomic perspective, to make it, to make it carry over into decision making on the field level, that's really where what those microbes do becomes the important, the valuable piece of data to look at. So that's where the second step of the process comes in, where we take that biodata, run it through a machine learning based process that our data science team has engineered over the last 6-7 years. And they basically match up species profile or species taxonomy of each microbe detected to the functions those microbes perform in the soil based on full genomic data that we've curated in that machine learning based process. And that's where we deliver the functional reports in the back end. And this is looking at specifically factors like nutrient cycling, biocontrol, disease, abiotic stress tolerance, that microbes can induce in crops. And we'll go through some of the details there, of course, when we get to the case study. But ultimately, the functionality data is what we deliver on the back end to help understand how product biostimulants, biofertilizers, also ag inputs as well as management practices, how it influences the microbiome and moves these populations towards healthier, more plant growth promoter networks of fungi and bacteria. So trials is really our more scientific approach to understanding product inputs and management practice impacts on the soil microbiome. And really, as BeCrop Trials, we, instead of just offering individual tests, this is where we really go into these projects as a partner to understand the end results. We get a lot of data in trials because we're setting up kind of like a typical university level or academic crop trial. In BeCrop Trials, we're setting up control and treatments to deliver very robust results, and we work together with our collaborators to make sure that we're honing in on really the big research questions and sort of what we can validate or prove as far as changes in the microbiome. So with BeCrop Trials, the idea is, we have a lot of folks who are using BeCrop tests, just individual ad hoc tests as an agronomic tool, often testing, fields that are struggling as far as yield or productivity, or sections of, golf courses that may be struggling or maybe are looking for biological product adoption, or folks who are doing like comparing a, side by side or a strip comparison of a product. But BeCrop Trials is where we take not just a single treated and control sample or a single before and after, but look much deeper at product performance, and we control for some of the variability that we expect to see in the field as far as climate variation, the natural microbial changes in the soil. And this allows us to answer some much higher level research questions about product performance. We can look at factors specifically like disease evolution management, understanding how products influence levels of soil-borne bacterial and fungal diseases. We can look at, understanding how products influence yield through improvements in soil biological functionality, understanding where inputs may perform better or worse at different application rates, different application timings and practices, as well as how products perform in conjunction with other variables in a management program. So one of the things we may look at is whether a product performs well or performs effectively when it's mixed with, say, a biological mixed with a fertilizer or even a fungicide. We can test efficacy as far as how they affect input performance. And what I think is really a key element is understanding the effects of biostimulants, other biological inputs across location over time. So our trials are typically done over the course of a season or even multiple seasons. And we've done some multi-location work with Kevin's team at Replenish Nutrients and seen some really cool patterns and trends play out as far as what benefits of Rebuilder and their other products are very consistent, and what benefits may vary between cropping systems, between locations. And we see that a lot. Just like the same, you wouldn't use the same exact fertilizer program or the same exact crop protection program in different regions. It's going to depend on what what nutrient deficiencies you want to address, or what pathogens or pests are an issue. With biologicals, we see that their performance also tends to vary across locations, and you may be able to better place and understand where they're going to help address deficiencies by understanding their modes of action. So that's what we try to address with BeCrop Trials. This slide, no need to go into a ton of details on trial design. This is just a basic overview of sort of how we set up these trials. We have, in each case, we always have sort of a minimum level of minimum needs as far as experimental design. And the three main needs are that we need a control and treated zone. So we need to be comparing, like in this case, we had the fairway that was receiving the rebuilder as part of the program, and then that other fairway that Kevin mentioned that wasn't receiving it. So we need a comparison, and then we need also to be able to take samples over time. So at least 2 time points to see how the soil biology varies over time. And then the third requirement is that we need replicated samples. So at least 3 samples, triplicate sampling at each time point and treatment. And the importance of that is having replicated sampling allows us to see how much variability there is in these microbial populations. So if there's this lets us run statistical analysis and have an indication of how confident we are that the change in the microbiome is driven by the product, versus maybe if there's a lot of variability in the results, maybe it's a little bit more likely that the weather, climate variables, natural variation in soil type and other factors may have led to some of the changes. And the basic way that we deliver the data, we compare these changes over time in the treated and control zone, and run what's referred to as a delta-delta comparison with the basic equation on the bottom. So we're looking at the change from the treated, the first time point of the treatment, T0, to the second time point, T1, and then looking at the change in the control, and basically subtracting the control from the treatment, or looking at the difference in we're looking at microbial functional groups and the percent difference in those functional groups in the treatment. So if we see a positive percentage in, say, nitrogen release or phosphorus solubilization in those functional groups of microbes that we look at, then we're indicating an increase in say phosphorus solubilization relative to the control when we see a positive number. So that's basically what we're doing, is comparing the percent change in each of the microbial functions we profile throughout the course of a trial. And ultimately, we deliver the data in a simple PDF report. It is quite, we have about 36 different microbial indicators or metrics that we look at that we're comparing between the treated and control. So it's about a 10-page report. And we also, there's a lot of data there to unpack, of course. We have, 2 different larger sort of categories of microbial functions: nutrient cycling, and then plant health. And of course, plant health is where we look at disease risk, stress tolerance related functions performed by microbes that help crops deal with biotic, abiotic stressors. That's where we look at biocontrol, the beneficial microbes that can suppress insects and pests. So there's, across those 2 categories of nutrition and plant health, there's a lot that we look at. So in addition to the PDF reports we provide that break down and highlight the biggest significant changes we saw associated with the treatment, we also provide a tailored PowerPoint going over the agronomic implications of the results. And our team prepares these deliveries to really dissolve down the data to the big picture takeaways, where we saw either maybe strong or maybe weak product performance. And especially with Replenish, which is, we're looking at with, Rebuilder, specifically a phosphorus-focused biofertilizer that has some other components, has the sulfur, and also has the organic matter or the proprietary compost. So multifaceted product, but as a biofertilizer, we kind of have an idea of what to look at. So that's what we focused on in the case study. And of course, with any trial, we try to, in our PowerPoint deliverable, highlight sort of what sort of research questions were we looking to ask, and what sort of product claims, or what sort of expected benefits of the product are we able to see the data validate and proof out as we look at the report. So I'm going to try to speed things up a little as we go into the case study, but really, as far as this just ties together what we're looking at as far as trial and ag input effects, looking at yield, looking at how it might relate to crop quality and risk management, looking at resilience of the microbiome and crop performance and understanding, how performance changes of a product throughout the season. So now to get to the fun part, the case study, and I definitely want to leave a little bit of time for Kevin and Mike to add any maybe practical insights or in-field observations that relate to this. But we're going over the BeCrop trial results for the Indian Springs Golf Course. So just the fields that Kevin showed some pictures of. This is the data behind the 2023 trial that was conducted there throughout the summer. So this is just the basic design, looking at a control and the rebuilder treated fairways throughout the season. So we have those two treatments, and then we had 4 time points throughout the season from May through to July. So a good portion of the active warmer growing season accounted for. And we really had, based on our previous work we've done with Rebuilder, and also what Kevin and his team, their understanding of their R&D process and their strong understanding of Rebuilder as a product. The 2 hypotheses we looked at really fell into 2 main focuses. One is nutrient cycling. So we expect Rebuilder to improve soil biological nutrient cycling, particularly phosphorus, being that we're introducing phosphorus in a more bioavailable form than a lot of your high salt index fertilizers, but looking not just at phosphorus, other nutrients as well. And then the second hypothesis that really fits more into the plant health section is, and this is the benefit we've seen in Rebuilder in canola, lentils, wheat, I think, and a few other crops we've trialed with Kevin's team previously. We expect that Rebuilder will increase populations of microbes that support plant stress tolerance. And this is, we look at microbes that produce secondary metabolites and other beneficial compounds like plant growth regulators, some of those vital hormones that Mike touched on. So we anticipate that Rebuilder would have some beneficial effects there as well to help the turf deal with some of the stress that it is, of course, going through it in many cases to more extent than a typical horticulture or agricultural crop. So this is just the overview of the results, and we have a cover page on the right of the trial where we have a very simple highlight of which metrics we saw major improvements in, and it's based on a simple star scoring system. We have the scoring legend listed there, but just for the sake of time, won't go into too much detail. But basically, the more stars are scored in each of these functional biological categories we look at, the better performance we saw in terms of the increases in those microbial functional groups in the Rebuilder treatment. Really, we saw big improvements in phosphorus and potassium pathways. Phosphorus, you'll notice in the report, had the 3-star rating, our highest rating as far as statistical significance and % increase in those phosphorus solubilizing and mineralizing microbes. So that was, of course, a big significant result we saw, especially in the early time points of the trial. We also saw some improvements in carbon cycling or decomposition. Organic matter release is the term for the metric we look at there. So those are the microbes that are able to break down like the lignin and cellulose, other complex carbons and other organic carbon sources that might be present in that actual Rebuilder, in the organic matter that's present. So that, of course, those are the microbes that are helping build organic matter, and that of course, we know organic matter is pivotal to water filtration and to an effective balance of water retention and versus leaching or runoff. So that we saw improvements there as far as carbon goes. And then stress adaptation on the bottom, we see salicylic acid and exopolysaccharide production, really small text on the bottom, but we also had some of the hormone and stress tolerance metrics that really showed major improvements, earning several stars for the increase in those functional groups. And then a few disease risks declined as well, including fusarium. So that was something we touched on a little bit, is when you improve some of these, this improved biological functionality, one thing that we can measure with BeCrop technology is levels of pathogens. So we actually saw declines in the levels of those pathogens and some of their associated risks. The next slide, this just goes a little bit more into the details of how many favorable or adverse changes we saw across the board in this trial. So the numbers for listed under the plus sign and the minus sign represent favorable, where we saw an increase in nutrient cycling, or an adverse change where maybe we saw a decrease in a nutrient cycling or increase in a pathogen. And this is not all the metrics. We have we have a few others we look at, like biocontrol, nitrogen, and micronutrients. But this was really the highlights of our research questions, or where we hypothesized Rebuilder would perform well. And we see it carbon, phosphorus, and stress adaptation, we really saw a lot of favorable changes in the data, and overall quite a bit more than we saw adverse changes across. We always expect to see some adverse changes, but in this case they were very minimal. So the next slide will just kind of touch on the report. This is just going to have, if you could just, maybe it's not showing up, or there we go. Yeah, so this slide just hits on some of the excerpts from the PDF trial report for the golf course here, and we have in the top just a piece of the carbon section. We look at several microbial functions involved in cycling each major nutrient: carbon, nitrogen, phosphorus, and potassium. And we see in our report excerpts on the right any metric highlighted in orange, and then we have the percent metric, the percent change listed there next to the bar. Any metric highlighted or outlined in orange is showing a statistically significant increase, so meaning we're very confident it was the rebuilder that was the variable driving that change. So carbon, again, we saw an increase in organic matter release throughout the season. This may be owed to those microbes that are present in the rebuilder, inoculation, or the carbon source from that organic matter. Maybe a combination of both, but throughout the season, at all three experimental time points, we saw that organic matter release was significantly higher in the rebuilder than the control. And same for phosphorus, where we saw 6 overall total benefits. A few of them here listed were in phosphorus solubilization, microbes that can help unlock phosphorus that's bound up in inorganic form, and organic phosphorus assimilation, microbes that help unlock organic phosphorus in the soil profile to make it plant available. And then stress adaptation, the last excerpt on the bottom at 12, we saw 12 favorable changes there. And a lot of those were in the microbial functional groups that produce metabolites like ACC deaminase, salicylic acid, and other beneficial plant hormones that microbes can produce and essentially donate to the plant through the root system. And that can help, especially in the case of turf, where we're talking about a uniquely stress-induced environment with all the mowing, a lot of very sandy, low organic matter environment, and potential for drought or very low levels, ideally lower levels of water use to not have to have excessive levels of irrigation. So we saw across this is just 2 of the metrics in the stress adaptation category that improved at all 3 time points, but overall we saw 12 improvements there as far as how microbes can help that turf deal with stressful situations. And some of those metrics also relate to to the soil aggregation. We look at microbial functions involved in improving the aggregation and texture of the soil to help induce water infiltration and overall, improved texture for root penetration. So that was where we also saw some notable improvements there that I think carry sort of align with what you saw in the field over the course of this trial, Kevin. So this just sort of ties together conclusions of what we saw, and in the hypotheses addressed here, we have just the same 2 hypotheses we're looking at. Expected to see improvements in nutrient cycling, particularly phosphorus, and we did. So that was validated. And then we expected to see improvements in these microbes that improve plant stress tolerance, and that was another one that we saw pretty resoundingly with those 12 favorable changes over the course of the season in that functional category. So really the claims, to put it into sort of the more tangible agronomic perspective, in the green box at the bottom, we have the benefits that we saw throughout the study. We saw the ability of rebuilder to promote plant availability and uptake of phosphorus and other nutrients as well, facilitate the sequestration of carbon through improved decomposition, aid in plant stress response to biotic and abiotic stressors, and mitigation of soil-borne disease risk. And one thing that we didn't really touch on is the indirect connection. Those microbes that help produce plant stress tolerance inducing hormones, that can play a role in helping plants prime their natural defense system. Plants that have the microbes helping provide these phytohormones and other beneficial metabolites, those plants are better prepared to produce their own compounds that may make it unappealing for an herbivorous insect to take over and start feeding on them, or for nematodes, other plant parasitic pests in the soil. So that was kind of what we saw, and that's related to the stress tolerance section we looked at as the kind of maybe a contributing factor to the reduced levels of grasshoppers. I think you mentioned, Kevin, as far as the case study. So I will stop here. We had a lot to get through, and I want to make sure we also have a little time for questions in the end. But want to open discussion with Mike and Kevin to any other observations you might want to touch on, or just any other discussion as far as these results we saw in the soil microbiome compared to your experiences with using Rebuilder and other regenerative soil biology focused products in the field.
I think the results definitely show what we've been seeing, like you say, Gus, with all our previous work with you guys and other research trials that we've conducted is, yeah, I mean, they're consistent, right? So it's a consistent thing that we're seeing, and it's attributed to, again, the biology and what's going on with the products that we're developing. So again, we're further developing, increasing the biology in our products and injecting more to kind of supercharge some of our products. So that's kind of coming down the line. We've been working with other companies on different biologicals to incorporate into our granules. So I think that's something that we'll see an increase in release of nutrients and response. Some of the research that we've done in India with some of these products that we've tested show a huge increase in nutrient release and yield potential on some of the stuff that they've done. So yeah, definitely what we saw at the Indian Springs golf course is kind of typical of what we've been seeing elsewhere. So, but again, it's that build up that we need to be able to get that biology kicked off and moving the system back to a natural state again, I guess.
Yeah. Excuse me. What? Hold on a second. Boy, bad timing for me in this head cold, but what we're seeing is we put all the pieces of the puzzle together for guys, and once we started doing this, we could start associating some of the changes that we see in the levels of amino acids within a plant and the levels of plant growth hormones within the plant or on the leaf. And by doing that, it's teaching us as well. I mean, if you were to pull up amino acids in a web search and start reading about it, all these scientists say, oh, well, we're rapidly growing, and they say this particular amino acid may do this, or we believe it does this. There's still so much more research that needs to be done, and we believe we're lending to that research when we gather all the data, and data doesn't disappear with us and with our customers. It's always on their spreadsheets. So if they're trying a product and the sales guy comes in, because everybody that walks through that door has the silver bullet. And so if they're trying a new product, they should find out what exactly that product is designed to do. And then we can track it through that plant with our plant tissue testing to see if it actually is doing what it's supposed to do. It could even do more than it's supposed to do. But I don't think that the science it's never going to be all the way there. Let's put it that way. And the growth I've seen in 7 years is probably more growth than happened in the 2,000 or 3,000 years prior to the new sciences that are out there. So, but that's, to me, it's about proving what you're doing is working, why it's working, and to do that, we have to have all this data to be able to diagnose problems. And again, once we started seeing that microbial side of the equation, a lot of lights went on for us.
We have a lot of great questions that are coming in now, so I think this would be a really good sort of natural progression into some of the things we've been discussing, and a lot of questions, a lot of really practical based ones that I think Mike and Kevin, you guys can kind of speak to from experience in this trial and other cases in turfgrass systems. But Sunny, anything else to mention, or you want to jump into questions now?
Let's jump into some questions.
Okay, we just had, I'm going to repeat them, read them off just so everyone can know what we're addressing, but I just had a really good one pulled up that one of the audience members asked. So what is the best, and this is probably, Mike, more in your wheelhouse. This is, I think, a top of mind of probably a lot of people tuning in for this call, and anyone really interested in the applications of biological products in turf. So the question is, what is the best way to approach a golf course with products that are microbial based or maybe biostimulant based?
Well, it's all about the carbon and the sugars. Well, sugars and carbon, all about carbon, really. And the way you would approach it is like I said, you find out what that product's supposed to do, and then through a series of tests of sending samples, and part of our program is guys send us samples every week. We make adjustments to their nutritional programs based off of that data. And we're gearing up to, because I can see where amino acids and products like that and plant growth hormones are going to have specific amino acids, specific hormones for specific problems. Really, the only way you're going to know that's working is obviously a visual. If you see something visual, that's going to work. But doing the testing, taking the time to do the soil test, and then and the biology test, and the tissue test, and the water test, and sometimes superintendents, your water's so bad that you're behind the 8 ball as soon as you walk on the property. So.
Yeah, thank you, Mike. We have a bunch of great questions and a few minutes left, so I'll try to key in on some of the good ones that we've had so far. I can start on answering this one and then turn it over to Kevin and Mike for any additional details you want to add. So the question is, once you have field data, what's the process for addressing a deficiency of certain organisms? Say, for example, potassium solubilizers are missing, based on a BeCrop test. What's the step-by-step process for getting them back into the soil and ensuring they survive? And I don't think that there's any cut and dry, silver bullet sort of scenario there, but I can kind of give my experience from as an agronomist at Biome Makers. We don't specifically prescribe any products. We've maintained the role of an impartial, white glove lab. So, but we do provide general recommendations, and there's a lot of routes to go. Of course, then we give general guidance and then connect folks with biologically experienced crop consultants like Mike and other retailers or crop consultants who have a good understanding of what biological products will work in a specific field scenario. But in many cases with potassium, there's a lot of increasingly more products out there for both phosphorus and potassium that are solubilizing inoculants that contain bacteria or fungi that can solubilize them. And then you also have the biofertilizers like Rebuilder we saw here, can improve the biological mobilization or the ability of the microbes to unlock phosphorus while introducing bioavailable low salt form of phosphorus. And then Rebuilder contains the compost or the organic matter to help feed those microbes a little bit too. But then there's also, as Mike has alluded, sugar-based products and other biostimulants, humic-based products, and other sorts of carbon sources that can also help invigorate those microbes to unlock potassium. So it's not a one-size-fits-all solution, but something we can provide the data at Biome Makers to diagnose those solutions that then a trusted crop consultant or biological biostimulant product expert can help address and match a grower, agronomist, a superintendent with products like Rebuilder, biofertilizers that can address that issue. Anything to add to that?
Well, from my standpoint, you got to be patient because it doesn't happen overnight. And
yeah.
I had a microbiologist told me, he was the head agronomist for Cargill for many years, and he said that if you started with 2 microbes and the earth turned into a ball of carbohydrates or sugar, within 25 days the earth would be completely consumed. So what that tells me, if you keep food sources down there, you can increase those populations. But what we've got going on now with Kevin's fertilizer and a lot of these inoculated fertilizers is we have a way to build the right population. And that one of those books I read, you know, they'd ask the last question in the book was, you know, are you afraid that you'll release some kind of super bug and wipe out the human population? And, you know, if you think about it, maybe that could happen, but everything is researched so much, but you got to be patient because so many guys will take it in and they'll use some product from a company. We know what we're targeting because he's telling us, I'm not getting deep roots, I'm not getting this. And so we know what he's targeting, and we can look at the data like you guys do. You analyze all your data, Gus. And but that's all I got.
Yeah, I think, you know, guess what?
I do apologize, people. If this would have happened tomorrow, I'm a little under the weather.
I think, yes, you know, part of like we're seeing some of that potassium solubilizing in our is because we've got a multitude of, like, we've got phos solubilizers, sulfur, potassium in all our products. So, and again, we're enhancing those as we're developing our products. We're increasing the amount that's in our product. So you're going to get a lot of that in all of our products that we have. So.
Yeah. So you're adding the solubilizers, but you're also offering a soluble form of phosphorus and sulfur and some of these other trace nutrients. And that, thank you, Kevin, because that actually addressed one of the other questions we had, which was asking about the components, biological components of Rebuilder, of that the biological component as far as the activated compost. So we're just above time now, so unfortunately, I don't think we're going to have any time for other questions, but we'll get responses out and reach out to everyone we haven't been able to address, and turn it over to Sunny now to conclude things. And thank you all for joining.
Yeah, I'll keep it quick here since we're over time. Yeah, thank you all for joining today, and of course, huge thank you to Kevin and Mike for joining, even being under the weather. Really appreciate it, and you both sharing your knowledge and experience with us. And Gus, great work as always, going through the results of that trial there. And yeah, so thank you to all of our participants. And as Gus said, we got some amazing questions, and we'll be reaching out to make sure those get answered. And yeah, so thanks for joining. We look forward to connecting with you all one day to discuss soil biology in greater depth.
All right. Thanks,
Thank
Sunny.
you.
Thank you, everyone. Take care.
Take care.
Thank you.